Thank you all for joining us for our June National Vaccine Advisory Committee meeting. We will now return from our break and we will start our next session. Our next session is Innovations for Immunity, Infants, Immunocompromised Persons, and the Elderly. And this will be a true hybrid session. We have 2 of our presenters remotely and 2 in person. The development and approval of scientific breakthroughs has opened new doors to tailor vaccines to benefit special populations, such as infants, elders, and those who are immune compromised. Our session will have 4 speakers. First will be Dr. James Main from the Pharmaceutical Research and Manufacturers of America, followed by Dr. Greg Poland from Mayo, Dr. David Dowling from Boston Children's Hospital, and Dr. Nicholas Wolgemuth from Kansas Health Science Center. Dr. Main, if you're on, you have the floor.
I am on, and thank you. And I want to thank the committee for the invitation and the opportunity to comment to you today. My name is James Main. As noted, I'm the Vice President of Science Advocacy at Pharma. I lead the science policy portfolio at Pharma, including issues such as pandemic response and preparedness, diversity in clinical research, and modernization of medicines development. Prior to Pharma, I had a 30-year career in pharmaceutical industry, worked in all phases and stages of research and development, including in infectious disease. And prior to that, my training was in clinical pharmacology and toxicology. Today, I'd like to make comments in 4 general categories. I'll keep my comments high level in the interest of time, but welcome questions. First category is a snapshot of the vaccines in development. Where is the industry now? What does the profile look like? Second will be some comments on the industry pandemic response that I think will be relevant to the topics for discussion today. Third will be exploration of opportunities and challenges that are currently encountered, and then fourth, a little bit of commentary on the potential future state. So in terms of a snapshot of vaccines in development, we maintain a tracking tool that does a real-time estimate of different types of medicines in development. Under vaccines, the number currently is approximately 300. 300 vaccines in development at all stages. That includes both preventive and therapeutic vaccines, but does not include COVID vaccines, which I'll comment on separately in a moment. COVID vaccine, of course, historically in that process has been a real game changer. There is a range of disease-based or therapeutic area-based research being conducted at vaccines, again, combining both preventive and therapeutic. That includes, in ranked order, infectious disease, oncology, allergy, autoimmune disease, including diabetes, respiratory disease, and GI diseases. Regarding our pandemic response from industry, there currently are over 300 unique vaccines under study. That includes, again, all phases, preclinical, early, and late clinical development. These 300 vaccines represent a wide range of different approaches to both the identification and the development of a vaccine. There are a number of different approaches that have emerged as being highly successful alternatives for rapid response in a pandemic setting. These include platform technologies such as mRNA technologies, such as the Pfizer Moderna vaccines, DNA-based technologies, such as the Novavax spike protein approach, and adenoviral vector platform technologies such as the J&J AdVac platform. These platforms have really revolutionized the ability to rapidly go from the identification of a infectious threat through rapid sequencing to the identification and initial development of a potential investigative vaccine. This also has tremendous implications both for pandemic preparedness for future pandemics, but also for our overall countermeasure strategy as a country. What this has resulted in, the use of these platform technologies has resulted in a huge speed up in the time that it takes to go from that initial identification of a threat to the identification of an exploratory vaccine. However, and now we're moving into opportunities and challenges, that speed up has not been reflected to the same degree in the speed of clinical development of new vaccines, in the speed of manufacturing of new vaccines, in the speed of identifying correlates of protection that are necessary to evaluate new vaccines, in the speed of regulatory pathways, or in the speed of the regulatory review process. So tremendous progress made, but it's been progress made in only part of the process of researching and developing a new vaccine. Another challenge that I want to highlight, and I hope there'll be further discussion of this by the committee, is that challenge of vaccine equity. This is a large and growing issue that affects across the spectrum of vaccines, but was particularly acute and particularly visible during the COVID pandemic. The impact of vaccine equity is felt and observed at all levels of research and development, including clinical research and diversity in clinical trials, including the development and the use of distribution channels, including economic barriers, those that affect healthcare provision and access, and of course communication and communication barriers and mistrust. A tremendous opportunity and tremendous need in that space to improve the equity that's present in the development and the delivery. I'll close with making just a few comments on the future state of vaccine development. As we've seen, COVID has resulted in a remarkable uptick in the activity in industry and elsewhere when it comes to research and development of vaccines. From our tracker and from my conversations with the leaders of research and development of our member companies, this includes a significant increase in both therapeutic vaccine R&D and in infectious preventive R&D. The resulting increase in learning has been made possible by a multi-stakeholder, multi-partnership approach that has been taken and has proliferated again on the heels of the COVID pandemic. This has led to an increase in pre-competitive data sharing and information exchange within the vaccine community. This has led to an increase in creative and collaborative research programs such as umbrella trials. And this has resulted in the sharing even of manufacturing capabilities and resources. Which brings me to manufacturing, which I'd like to highlight again as a challenge that is in need of further both investment and research and development. Manufacturing and supply chain related to vaccines are a critical component of the overall development and delivery of a new vaccine product, and one that again has not enjoyed the same rapid advancement from a technology standpoint as has been seen in other areas of research and development. Regarding supply chain, it is critical that a global approach be taken and be considered, and that we don't restrict the supply chain or the flexibility or the redundancy that is built into these supply chains in any way. I'll offer by way of example the importance of the ability to move rapidly to source nucleotides to used in the manufacturing of the mRNA vaccines. There were, of course, few concerns regarding the ability of the supply chain to meet that. But I think we've all seen the remarkable success that resulted from effective partnerships and effective flexibility built into those supply chains. I'll stop my comments there in interest of time and in deference to some other fellow panelists. Thank you.
Thank you, Dr. Main. Our next presenter is Dr. Greg Poland from the Mayo Clinic. Dr. Poland, your slides are up. You have the floor.
Thank you, Bob, and good to see you. And thank you for the invite to ASL Interpreter 1. I apologize if I move quickly, but I'm happy to come back and fill in the blanks for you. So I was asked to talk about some of the work that I've been doing the last quarter of a century toward vaccinology 3.0 and personalized vaccinology. Next slide. My agenda is to define and discuss the evolving field of vaccinomics, a term you may or may not have heard of, discuss inhibitors and barriers, and through the above, really enlarge your perspective on a new direction that vaccinology is finally taking. Next slide. A trigger warning. Many in the audience may well have developed cognitive antibodies against the innovations and barriers I'll raise. And the effect of those high affinity antibodies is to bind to intelligence receptors, sometimes resulting in neutralization of innovation receptor networks and resulting in a continuing desire for the status quo. At least that's what I've found as I've discussed these concepts over the years. Next slide. So vaccinology 1.0 was really a simplistic isolate, inactivate, inject paradigm, and many good vaccines were developed out of that. Next slide. Vaccinology 2.0 involved recombinant technology, new adjuvants, subunit vaccines, but it's still operated under a post-World War II paradigm of everybody's at the same risk, so everybody gets every vaccine, same number of doses, and for the most part, the same dose amount. And in addition, was focused exclusively on prophylactic vaccines and predominantly on childhood vaccines, despite demographic changes that warrant the opposite. Next. Edward Jenner's first paper, by the way, on the protective effect of cowpox against smallpox was rejected with the words, "It was too revolutionary and simply not possible." Next. So if innovation were a crime, would the evidence be sufficient to convict vaccinology of being a highly innovative, cutting-edge science? Next. So the future of vaccinology, I'm going to focus on the bottom two in blue, is really toward vaccinomics and systems biology approaches and the development and integration of personalized or individualized vaccinology into medical practice, the same as the rest of our medical practice. Next. So who are the inhibitors? Well, primarily, ironically, vaccinologists and clinicians who don't understand at depth immunogenetics, systems biology, and immunology. And therefore, sort of the natural human way of dealing with that is to be uncomfortable and to tend to reject or stall it. And this is a difficulty with parting with this mindset we've all grown up with and has been heretofore relatively successful. But Kuhn was right. Next. Who said new paradigms come slowly because myth, error, and superstition cloud our ability to see the data. You all see this in reviewers. We wrongly believe scientific progress is a process of linear accretion of knowledge, that science is predicated on the belief that the scientists themselves understand what the world is like. And most importantly, the resistance to fundamental novelties, because they're seen as subversive to our firmly held beliefs. Next. Deming said, after all, it's not necessary to change because survival is not mandatory. Next slide. And this really introduces a paradox. Our goal when we give a vaccine is to induce protective immune responses in 100% of the population. That never happens. And that's because of pathogen variability, human immune system or host variability, and a lack of variability in how we deliver vaccines. Next. Just to give you a glimpse at host variability, is there's about 10 to the 12 different products encoded by the hundreds of immune response genes. Just in HLA haplotypes alone, the theoretical number is greater than 10 to the 13, yielding essentially an almost infinite diversity of human response capability. Next. So the easy vaccines, I think most of us would agree, have been developed. The remainder involve highly complex, variable, hypermutable viruses and complex bacteria, parasites, and fungi. Next. So why do immune responses to biologics vary? And they do quite substantially. And what explains this heterogeneity? And how should it inform policy and development of vaccines? Next. So let me take you into the future. Next. So in my lifetime, certainly the lifetime of my grandparents, we've gone from holding people down to chop their leg off to robotic surgery, from simple Mendelian genetics to, if you come to the Mayo Clinic today with resistant hypertension or depression as two examples, you'll be genotyped. And we've had stunning success with directed or individualized treatment via that mechanism. Next. So I'm hoping tomorrow we'll be more clever. We will have a personalized approach with directed vaccine development, that we will have tiered risk and vaccination approaches, despite the minimal complexity that introduces to the clinical office, different ways of administering vaccine, different adjuvants, and we'll pay attention from the change of a pyramidal demographic structure to a pillar demographic structure and develop adult vaccines as our primary focus. Next. So what does personalized mean? It could mean individual, as they do in cancer biology, but for the most part, it may involve different vaccines or routes by gender or by ethnic group or subpopulations with specific definable genetic polymorphisms, or subpopulations who have specific diseases, receive specific therapeutics, or have different immune states. Next slide. So vaccinology 3.0 then becomes a paradigm not of isolate, inactivate, and inject, but one of discovery, validate, characterize, and apply. And this is really where we get into vaccinomics and systems biology approaches. Next. So it starts with something that my laboratory developed some time ago called the immune response network theory, where the response to a vaccine is the cumulative result of, and this is key, non-random interactions with host genes, epigenetic phenomena, metagenomics, the microbiome, gene dominance, complementarity, epistasis, co-infections, and other factors that occur within the system as a whole. So again, a systems level approach rather than the approach most clinicians are comfortable with, which is a reductionist approach. Next slide. This then wraps up into a term we coined called vaccinomics, which is the integration of systems biology with the immune response network theory, immunogenomics, and immune profiling and functional studies in order to understand and predict vaccine-induced immune responses, protective or aberrant, and uses that information to reverse engineer new vaccine candidates and to drive individualized vaccinology. And there is now, I've just listed some of our papers, there is now a rich literature on this. Johns Hopkins University just did a scoping review, and it's just an overwhelming number of papers now. Next. So what does this look like? Well, to start with, it involves high-dimensional approaches to assess responses to vaccination, and then entails bioinformatic approaches to interpreting those data. Therein is why clinicians are uncomfortable with this, because they're not trained in this, of course. They're not comfortable with it, and they're unprepared for 21st, much less 22nd century medicine. Next slide. And so we take these high-dimensional, multi-parameter immunogenetic microbiome studies, use statistical analyses toward data integration, computational modeling, and we're able to develop novel correlates of immune protection, advanced understanding at the systems level of the biologic response to vaccines, predictive biomarkers of vaccine response, something pharma is particularly interested in, early immunosignatures of adverse events, and as I said, reverse engineering toward directed vaccine design. And like all of science, this is an iterative cycle of discovery, replication, validation, and application. Next. So we predict, and indeed we're already in the era of personalized predictive vaccinology, where we will abandon a one-size-and-dose-fits-all, predict whether to give a vaccine based on the likelihood of response or need, predict the likelihood of a significant adverse event, predict the number of doses likely to be needed to induce a protective response, and design new vaccines. Next slide. Now, let me take you back about not quite 20 years when we introduced this. Here's what I got from colleagues. No one's ever proposed individualized vaccines. How do we know it can be done? Journal reviewers, if you can imagine, the author fails to demonstrate that vaccinomics has led to a licensed vaccine product. From an NIH reviewer, the applicant proposes a novel paradigm in vaccine development. And we're only talking in this grant about viral vaccines. But can it work for non-viral vaccines? Pending such data, this falls to a lower priority. Next slide. All one needs to do is wait, and in time, truth flies. This is an interview from Scientific American on our work, and they called it one of the most innovative scientific concepts of the decade in one of the hottest omics fields. Next slide. So if you look at the bottom right of the slide, this is really what all of this points to. In time, and I'm going to show you briefly that it's already happening, we will determine via this individualized approach in vaccinomics whether to give vaccine A, or whether, because of the risk of an adverse event, to give vaccine B, or perhaps an alternate dose or route, whether to give vaccine B because it's higher dose or adjuvanted, or whether we need to give different antigens because of primary vaccine failure and use vaccine D. Next slide. So has that already happened? Well, yes. We've already designed new vaccine candidates using this systems level approach, and you'll recognize that it is a directed rather than the current paradigm, which is rather empiric. We're beginning to explore more another field we coined a term for, adversomics, where we're understanding the genetic drivers of aberrant or autoimmune or non-immune responses to a vaccine. Next slide. So this future has already started. We have, in the way of thinking about individualized vaccinology, a disease model. For example, we give different doses of hepatitis B vaccine for those in chronic renal failure. or the age model. We give different doses, different amounts based on age, or different adjuvanted vaccines for the elderly, for example, with influenza. For when I started in the field of vaccinology 40 years ago, there was one and only one trivalent injectable influenza vaccine. We have some 9 different types now so that we can choose the right vaccine for the right patient at the right time. And a genetic model where increasingly we understand, for example, my group has defined SNPs in SLAM and CD46 genes, the receptor genes, that code for 3-fold reductions in antibody responses to measles vaccine and are helping us to define vaccine failure in the context of the most transmissible virus known in humans. Next slide. And so we have the beginnings of a personalized approach for influenza, for hepatitis B, for COVID-19 vaccines, just as an early start into the field of individualized vaccinology. Next slide. In the way of adversomics, and this is just the briefest smattering, where we're already beginning to define specific gene expression profiles that code for and lead to fever such that febrile seizures occur, or rashes, or lymphadenopathy. And these are important beginning steps toward understanding the harder to study because they're rare side effects. For example, we are starting on using these approaches to looking at myocarditis in COVID-19 vaccine recipients. And can we actually develop an immune signature that would reveal this person's at higher than average risk? Next slide. So the usefulness here is to understand immune signature profiles from a vaccinomics perspective and use these markers in personalized vaccinology to inform new vaccine development, to predict aberrant responses, and in the industry perspective as go, no-go markers. Next slide. So what would Kuhn say at about this point? Well, we make the mistake of requiring new theories to explain all the facts with which it can be confronted and reject those that don't. And new advances, frankly, he says, require that we must attract adherents away from competing theories. It generally, for something as revolutionary as the new biology in genetics and medicine, will require a new generation of healthcare workers who are facile with these concepts. Next slide. So after 3 decades of work, I can tell you in my own laboratory, we've defined polymorphisms of specific immune response genes that significantly influence immunity to measles, rubella, influenza, vaccinia, and anthrax vaccines. We've been able to predict and profile vaccine-induced immune responses that are not protective. We've used these to radically change the field of vaccinology, particularly to understand how vaccine antigens actually work to stimulate protective or pathologic immune responses and to identify new vaccine candidates. I think next and almost last slide. So I think we will abandon a one-size-and-dose-fits-all vaccine approach. Using gene signatures predict whether to give a vaccine based on the likelihood of response or on the likelihood of a significant adverse event, predict the number of doses likely to be needed. We can already do that with hepatitis B vaccine, and again, design or develop new vaccines. Next slide. So 2 rules for success. Next slide. Number 1, never tell everything you know. Thank you very much. And I think at the end, we're going to have questions.
Thank you very much, Greg, challenging us as always. Our next presenter is Dr. David Dowling from Boston Children's Hospital.
thank you everyone for the opportunity to present today. And my name is David Dowling. I'm a principal investigator of what I've designated the Adjuvant Discovery and Development Lab within the Precision Vaccines Program at Boston Children's Hospital. I'm also faculty at Harvard Medical School. What I'll try and do today is just building upon the presentation from Greg, which is a very nice broad introduction to some of the topics that many of us in the field of what he calls vaccinomics, and sometimes we prefer to call precision vaccines. And I'll try and firstly try and center a little bit the importance of focusing on distinct immunologically distinct populations, and then secondarily give a specific example of adjuvants. And so hopefully this will align somewhat with Dr. Hopkins' idea of trying to be simple and keep to an idea that's a good take-home message. Next slide, please. So one of the first things that we want to try and put across is that many factors impact vaccine responses, and they're not necessarily the same. We're just highlighting 2 or 3 or 4 examples here that I find are very important. Firstly, biological sex. It impacts individuals' immunological responses. Age is very important. Younger individuals, as compared to people across the lifespan, as individuals become older, or have different immunological responses. And we see this in terms of infectious disease susceptibility. Those at the extremes of age tend to be more susceptible to morbidity, and also there's comorbidity plays a role in vaccine responses generally. Seasonality is important for infection, the type of vaccines are given, schedule, the formulation, and there's population differences, especially around the world. Next slide, please. So one of the things that I want to try and highlight is the reason why we have distinct responses to vaccinology, partially building upon what Greg has mentioned, is that there are different immunological states, especially by age. One of the things that one of the ways we tried to highlight this, and this is a paper from a few years ago written by myself and Dr. Levy, Ofer Levy, who's the Precision Vaccines Director here at Boston Children's Hospital, is that different age groups have immunological states that are distinct for that population. For example, infants or young neonates in particular have a very distinct immunological profile that's designed biologically and from an evolutionary point of view to optimize their lifestyle or their immunological need and how they interact with the environment or other humans. And this sometimes is referred to as a layered immune response. As individuals grow older, from 1 month to 2 months to 1 year to 2 years to 10 years, the immune system changes. And this leads to distinctness. And so one of the things that we've been trying to highlight over the last many years, and Greg touched upon this, is that to better design vaccines, you need to first understand the immunological set point of different populations. And disease also plays a role. People who have various different disease manifestations may also represent with distinct immunological states. So it's a complex type of system that's constantly changing, and you have to adjust the science for these different populations. Next slide. So the phrase that we've often used for this is a little bit different. We sometimes call this precision vaccines. Many people define precision medicine as trying to tailor the treatment for the individual characteristics of the patient. And Greg is building on this type of idea where we take a slightly different approach, where because vaccines are mainly deployed at a population level, we should try and design immunization strategies for those specific populations. So it's taking into consideration the distinct immune state of infants, older individuals, or immunocompromised individuals, the overlap between those groups. Next slide. So one way of approaching this is building upon some of the ideas that we've mentioned already on this session, is where we can learn from natural immunity and the disease states that come from this. And this is from a review article from some of the postdoctoral fellows, including Dr. Parman and my team, where we can take inspiration from natural immunity and learn how to instruct better immunological responses to avoid the worst disease outcomes. And one of the ways we try and approach this is we tend to have a research cycle and an innovation discovery cycle that takes into consideration various different ways of evaluating vaccines or inventing new technologies. And this, firstly and foremost, involves trying to consider age or population-specific responses as in this middle cycle, and also taking into consideration new ways of evaluating vaccines, whether they're in vivo models of animals, in vitro modeling where we use human tissue samples in a dish, and some of the points that Greg mentioned, using systems biology approaches, systems vaccinology approaches, high-level technology, and new ways of understanding and generating hypotheses based on the tools. It also means being very aware and having mixed methods researchers that are accustomed to and knowledgeable of vaccine design from an antigen level, from a formulation level, and also how to deliver routes. This is challenging, but if you get it right, you can lead to some great outcomes. If you go next slide, please. So specifically, as an example, what our team tends to do is in this innovation cycle of precision vaccinology, we tend to try and discover new ways of developing vaccines using in vitro modeling systems that generally help us down select on technologies that will lead to better immunological responses by age or by function, using the most appropriate animal models, whether young animals, old animals, animals that have similar to as possible immunological responses or genetically modified to better replicate the human immune system, and ensuring that the human clinical trials where possible include as broad populations to make sure vaccine responses are replicated and as broadly applicable to different population groups. And so when you see this in the context of mRNA vaccine development, is that it was surprising and still somewhat suboptimal that infants and young children under the age of 5 are still waiting for a vaccine that hasn't been developed specifically for them using that technology because of optimization characterizations in terms of reducing reactogenicity or other safety concerns, or secondarily, the lack of immunogenicity is that the vaccine needs multiple doses to get to the level that you see in older individuals. And what's important, and I agree with Greg in this matter, is teams need to evolve and begin to incorporate new tools to help them get as much information from these systems as possible. We often call these systems biology approaches or systems vaccinology approaches, often using omic technologies, whether it's transcriptomics, proteomics, and metabolomics. The key thing that is to mention about these technologies is often they are helpful for generating new ideas and new hypotheses rather than necessarily always answering the most important questions. Next slide. What I want to try and do in this case is just focus on one technology that in some ways actually in my mind has been represented in the context of, for example, the response to the pandemic, but is extremely important for global access to vaccines, and this is the concept of adjuvants. So adjuvants are molecules, usually small molecules or particulates, or immunomodulators that can enhance the magnitude, breadth, durability of the immune response. And so this is from a review article. It's a nice graphical overview that I sometimes present, where on the left-hand panel on A, a non-adjuvanted vaccine, where you have a, say, for example, protein antigen that's injected intramuscularly. It's taken up usually by your immune system in the innate phase. The antigens are processed, and you basically have an interaction with T cells that leads to clonal expansion of T cells that also help antigen-specific B cell responses that lead to an antibody response. In a non-adjuvanted vaccine sense, often what can happen, though, is that you have a very narrow immunological response and it wanes over time. So many of us who work in this field of adjuvanticity are constantly trying to find new ways of coming up of vaccine formulations that will work in a way that enhances broadly immune responses, but does it in a way that also overcomes the distinct immunological states of individuals who have, or in early life, later life, or may have other immunological distinctness, such as a comorbidity or an immunological disorder. And in this case, usually what happens is an adjuvant can be added to a vaccine formulation and it can enhance the innate immune response. Classically, not always the case. Sometimes the adjuvants can directly interact with T cells and B cells, but generally they usually drive an innate immune response. So this is usually a precision inflammatory response, and you get a much broader T cell response and a B cell help from the T cells in the lymph node, and then you drive this long-lived and more broader immune response. And one of the reasons people are also interested in adjuvants is that they broaden the breadth of immunity, so you can reduce the number of doses. You can have a broader response that will deal with situations such as escape variants that we see in SARS-CoV-2. And these are all the advantages that come with in precision adjuvanted vaccine development, including improving the immune responses of populations who couldn't otherwise be vaccinated. So, for example, the vast majority of pediatric vaccines in the world are adjuvanted with a substance called alum. because it helps enhance antibody responses broadly. Next slide. So as a further example of this, and I'll finish up with just this story of some of the work that our team had done during the early stages of the pandemic, where we put together a coalition basically of teams who are interested in adjuvants and vaccine technologies. And we solicited anyone who had interest in collaborating with us in this space. And we were able to take some of our own adjuvants that we had developed, commercially available materials, and also academic industry partners. And we set up a cycle of research where we studied the immunological responses using in vitro modeling of individuals who are older, because these are the most susceptible population in SARS-CoV-2 space, and also trying to model in vivo which formulations would be the best as a proof of principle for targeted vaccine development for vulnerable populations using older animals. Next slide. So one of the things we also wanted to do in this space was prove that we could develop vaccines that are relatively cheap. And this is also an important aspect in terms of having global access to formulations that many people may not have access to in an easy sense in terms of mRNA vaccines, for example. So we focused on protein subunit vaccines. Typically speaking, and historically, these vaccines tend to be cheaper, they're easier to make on scale, and they tend to have less historical issues in distribution globally. And the easiest protein and the best immunological target in this sense is the receptor binding domain of SARS-CoV-2. It's easy to produce, but it's very poorly immunogenic. In essence, if you immunize individuals with this protein alone, they won't make a very good immune response. So to overcome this and get the benefits out of a protein like this, you need to try and figure out an optimal adjuvanted approach. And so we set about doing some research in the space early in 2020, and published some work in this in the last several months. And I want to just highlight, just as an example, how this is important for targeted vaccines. Next slide, please. So here's an example of looking at the immune response to a screen of various different adjuvants, either alone or in combination in a mouse setting. So we took the RBD protein and we looked for it combined with 3 or 4 different known adjuvant systems. They're listed on the bottom left. And what you can see here in general, this is a response known as a surrogate viral neutralizing response that indicates how good the antibodies that a mouse produces may bind to a certain receptor that's related to the virus in question. And most of the adjuvants alone don't work well, but if you combine them together with alum, you get a distinct response. And there's one here that pops out as being most immunologically relevant. It's a combination of alum plus a toll-9 agonist called CpG. And this agonist led to the highest level immunologic response to this combination formulation. And so we classified this as alum CpG RBD vaccine as a promising candidate. Next slide, please. What's really interesting is during more than a year's worth of work, we did follow-up studies looking specifically in models of the age population. And what we found was something really surprising, is that much of the responses that you see with this combination have age-specific readouts. And so on the left-hand side, we're just indicating the total antibody response to RBD alone plus various different adjuvant formulations. And what you can see is that there's distinctness, so not all formulations work the same. Particularly interesting was that only one of these formulations was able to induce protective antibody titers in aged animals, and this was the combination of alum plus CpG adjuvant together. So this idea is sometimes what we call precision adjuvanticity, is that you have to do the research in those populations to try and figure out which is the best formulation to try and target. Next slide. And furthermore, basic mechanistic studies in vivo, we were able to figure out what the mechanism behind this type of phenomena. Generally speaking, what happens with a vaccine response is that you end up getting an immunological response in the lymph node, as we talked about previously, where you have the innate response driving adaptive response. In young animals, generally speaking, you have a phenomena in which you get helper cells. These are white blood cells, T cells called T follicular helper cells. They're a subset of cells that help your B cells make antibodies. Generally speaking, those are much more abundant after a vaccine response. What's interesting is, conversely, and that is somewhat unique to age, is that individuals and animals who are older tend to have regulatory cells that suppress this response. And only certain types of vaccine formulations can overcome those distinct responses. And that's what we found out was happening with this unique combination. Next slide. Interesting, one of the things that we were able to do to help us go down this path, and this happened concurrently with the research in the animals, is we're able to also study these different formulations using age-specific responses in the context of the human in vitro modeling. So we have a biobank that our team generates, where we have samples from all different ages of individuals, from very, very young up to very, very old. So some people samples have donated, and participants have donated biological blood samples, so start upwards of 100 years of age. And what I'm just showing here is an example of some of the studies that we can do in this space, where we can take the adjuvants alone in panel A, alum, CpG, or the combination of the two. And what we're just showing here is a representative radar plot looking for the ability of these combinations to induce cytokine responses that are mimicking the innate response that one would see in the human in vivo setting. And what's really interesting is the combination of these two formulations is indicated in the purple leads to a synergistic response. So when you add 1 + 1, you don't get 2, you get 10. You get a massive response. And what's most important is normally, as individuals get older, their immune responses wane, but this does not occur with this unique formulation. And so this is just a very precise example of how you can use the combination of these different tools that myself and Greg have mentioned to actually down select in terms of the type of formulations that might work best. It's not just necessarily about having the fastest vaccine. Ultimately, it's about having the vaccine that works best in these populations. And these formulations, if you go to the next slide, we recently published in Science Translation Medicine, just as a very simple example. And furthermore, these formulations laid the foundations for various different teams around the world to use this type of adjuvantation system in upwards of 7 different vaccine formulations that are trying to deploy both the RBD, the spike protein, and the combination of these adjuvants, because they work so well in all the populations to a point that they're similar to or even greater than mRNA vaccines, but in a way that may be upwards of 10 to 100 times cheaper in essence. So most of these vaccines are costing less than $1 to make, and they can be stored and transported much easier than other vaccine technologies. As one example, I think it's just worth considering. Okay, thank you for your time. Happy to take questions at the end.
Thank you very much, Dr. Darling. Our next presentation is by Dr. Nicholas Wilgemuth from Kansas Health Science Center. You have the floor, sir.
Howdy. I'm impressed you got my name right. I just want to basically thank Dr. Poland and Dowling for introducing a lot of the topics I'm going to be talking about today. So I should be able to get through some of this introduction pretty quickly. So most medicines and medical therapies that we think about are targeted against diseases, right? Targeted against disease, either bacteria, viruses, disease tissue. For example, antivirals target viruses. But importantly, when we think of vaccines, we sometimes think of them as targeting pathogens, but they're actually targeting the healthy immune response. And what's important in the immune cells that make up that immune response. And so what's important here is that humans and their immune systems, as talked about earlier in this session, vary widely by demographics and by individuals. And so why would we think that one size fits all would be appropriate for vaccines when we're not treating, just one virus, we're treating the entire immune system of diverse individuals. And as talked about, these are just some of the examples up here of age, immune history, sex, and gender that make up some of the differences in immunity that, in this case, when I'm going to be talking about, affect influenza virus vaccine efficacy. So one of those that is one of the best well characterized, one of those differences is sex and gender. Specifically, and I'm just going to focus here at the bottom for the vaccination part, but we know that males tend to have less adverse events following influenza vaccines. Females tend to have greater adverse events following influenza virus and other respiratory virus vaccines, but women also tend to have greater B-cell responses, greater antibody responses, and actually, when you measure vaccine efficacy in females, it tends to be greater than that in males. This is just a quick slide showing that in age, you also have this effect of the hormone levels dropping off with age, leading to likely, in part, some of those differences in immunity that we've seen and talked about by Dr. Dowling and others in this session about what makes elderly populations so susceptible to infectious disease and unable to mount effective immune responses to vaccines. Obesity is another one of these risk factors that's and you can see that when you look on that top left graph at the antibody response following a vaccination in an obese individual, you can see that the IgG, the antibody response, is roughly the same or maybe even a little bit higher compared to a normal weight individual. However, when you look at that secondary response, that immune response in the bottom left-hand panel, you can see that those antibodies, while there may be more of them, they're not that good. They're not as specific to influenza. They're not as specific to the epitopes in influenza that are protective. And so while obese individuals are able to make antibodies, they're not always the most effective and protective. And that's shown on the right here. And so this is all in vaccinated individuals, where you have on the far right, the very obese have a risk ratio of about twice that of the normal individuals for developing influenza-like illness. And again, these are in vaccinated individuals. The very obese still have that higher risk. And so, again, as has been talked about, many factors can affect this. Males tend to be harder to vaccinate than females. Elderly tend to be more difficult to vaccinate than the young. Pregnant women are a particularly difficult to vaccinate population. Their responses, they seem to be harder to protect, particularly to protect the outcome of the pregnancy. And then obese individuals are harder to protect than individuals with healthy BMIs. And so the answer here is, that differential immune responses to vaccines require different vaccines. And so this is just, I want to just briefly bring up this here to point out that we are already doing this. We already have some of these precision vaccines that we've talked about. Maybe not quite as precise as what Dr. Dowling's working on, but we know that high-dose influenza vaccines protect the elderly. They induce a greater immune response, with it, like the data on the left shows, and they also have an increased efficacy compared to standard dose vaccines shown on the right. So this is something that we are already using and doing to good effect to save lives. Now, adjuvants are another important aspect of this. However, when you look in obese animals, both adjuvants and high-dose vaccines don't seem to be very helpful. So if you look at this graph here, obese animals are on the right, lean animals are on the left, and vaccinated obese animals don't show much of an antibody response. However, once you add that adjuvant, that antibody response becomes much more similar to the lean animals. And so you rescue the antibody response, so those animals should be protected, right? So the lean animals, you vaccinate them, they're protected, they get a little sick, but then they survive. The obese animals get very sick and die, even though they had antibody levels similar to that of the lean animals. Now, what if we combine the adjuvants with the high-dose vaccine? And here's just a survival curve on the right showing that when you have a vaccine and an adjuvant at the high dose, you are able to significantly increase the time the mice live, but they still are susceptible to severe disease and death. And so while it's more protective, it's not protective enough to save the lives of the mice in this model. And so when we start thinking about protecting these diverse groups, we start wondering what's all in our toolbox. And this was talked about by Dr. Poland. We've got our inactivated vaccines, some cell-grown vaccines, live attenuated vaccines, and we've got different adjuvants and doses that we can administer. So when we start to think about personalized vaccines, we can start to think that for a normal health individual, age 6 to 65, an appropriate prescription for vaccine might be the standard dose inactivated influenza, whereas someone age 65 and up might be better off with the high dose influenza vaccine. And then we know from prior studies on LAIV that it is particularly effective in individuals that don't have much immunity to influenza. And so it can be, it has been recommended that in some non-clinical settings that children between the ages of 2 and 8 get the LAIV vaccine, the live attenuated vaccine. But what about obese individuals? Do we have anything for them? No. What about pregnant individuals? We just tell them to get vaccinated, but we know it may not work very well. And the immunocompromised, yeah, we tell them to get vaccinated, but they don't mount much of a protective response when we do vaccinate them. So what are the problems here? One is that not all of the demographics that we've been talking about have safe and effective vaccines. And again, these are demographic-based vaccines. These are not yet personalized vaccines, and that's getting more to what Dr. Poland was talking about. So when I think about the approach to take to get to personalized vaccines, what we really have to do is fine-tune our vaccination strategies and platforms, not just to the age, gender, body size of the individual, and not even just the genetics, right? The immune history of the individual, right? Everyone in this room has had influenza or been vaccinated, or most likely had it more than once. And that's going to affect how you respond to your next vaccine. And so starting to get to that, not just your genetic history, but your immune history, can better inform that personalized vaccine approach. Now, once we have all this data, and we have lots and lots of, we know that this person needs a less attenuated vaccine, this person needs a stronger vaccine, we need more platforms, we need more technologies, we need more dosing levels, adjuvants, like Dr. Dowling was saying, and routes of administration to basically get a greater diversity of ways we can deliver and administer these protective antigens. mRNA vaccines are going to be a big help here. In theory, you can generate multiple candidate vaccines and get each season, and so they will have a big role, I think, going forward with this personalized vaccine approach. And again, as Dr. Dowling was, as Dr. Poland was talking about, that in-clinic genetic profiling and, in this case, immune profiling are going to be the best way to assess what each individual needs. But as I mentioned, we have that problem of not having enough platforms. So I'd just briefly like to talk about how we can maybe get more out of the platforms we do have. So the live attenuated influenza vaccine has the HA and NA, the primary immunogens from the wild type circulating strain that you're trying to protect against. It's got its 6 internal gene segments, shown in red here, from the vaccine strain. And that vaccine strain was cold adapted to grow at cold temperatures, which resulted in 11 mutations in those internal gene segments, 6 of which have been linked to the attenuation of that virus. So it's 6 mutations that attenuate influenza in the flu mist vaccine that is administered annually. But LAIV has a bit of a problem. It was not recommended a few years ago, and that's because it's thought that the effectiveness was decreasing following the pandemic 2009 H1N1 emergence because of being overly attenuated. The pandemic virus was actually not that great at replicating in humans, so when you added those attenuations, it was able to replicate even worse. And so a normal attenuated virus vaccine will be able to replicate to nearly wild type virus titers and still induce those protective cytotoxic T lymphocytes, those CTLs, and those antibodies. But an overly attenuated vaccine virus will grow worse and potentially not be able to mount or stimulate a protective immune response. And then the worst case scenario, which is when you're administered an overly attenuated vaccine and you had flu the season before, you're going to have pre-existing immunity that knocks that already overly attenuated virus down even more. There's essentially no chance that that vaccine is going to provide you with much benefit. And so LAIVs, interestingly, can have that attenuation fine-tuned. You can reverse or add mutations that make it more or less attenuated. For example, you could add some of the mutations associated with the Russian LAIV to make it more attenuated. But in this case, what we were doing was trying to see if we could make it a little bit less attenuated. And so we studied the LAIV in human nasal respiratory cells, and we found that when you reversed this one mutation, this M2S86A mutation in the vaccine strain, and mutated it back to the wild type allele, the alanine, you're able to, as you can see, greatly increase the replication of that virus. And so the idea here is that that increased replication would mean this is less attenuated. And if it's less attenuated, it can replicate better. If it can replicate better, it can lead to a more robust immune response. However, another population I was talking about, women, are more susceptible to severe adverse events following vaccination. And so for women, a stronger, less attenuated vaccine might actually be contraindicated because it would be a vaccine that would already cause some problems, could cause even more if you were to decrease its attenuation. And so just reiterating what has been said throughout this session is that one size does not fit all. The first steps that we need to do are to design demographically based vaccines. We can do a lot of that with the currently licensed influenza vaccines that we have. But that next step is really going to be what Dr. Pullen was getting to, looking at the genetic profile, looking at the immunological profile of these patients in a clinical setting, and doing a quick assay to see if they have what genotype they have, to see what immune profile they have, to see what vaccine will be able to best protect them. But the 9 vaccines that we have aren't going to be enough for everyone. And so we do need to develop new vaccines. And like Dr. Dowling was talking about, we need more adjuvants and better combinations of adjuvants and vaccines to effectively meet the need here. And so hopefully I didn't go too long, but thank you.
Thank you, Dr. Wohlgemuth, and I want to thank all the members of our panel. You've really challenged us. As I think through these presentations, and I think about my role as a frontline clinician and patients coming in to see me and thinking through personalized vaccinology, I see a push me, pull you. On the one side, trying to move toward personalized vaccinology where we reduce the risk of adverse effects, optimize vaccine-induced immunity for individuals. But on the other hand, if we don't vaccinate our patients when they're in the office, we miss an opportunity and potentially don't get them vaccinated. So, I'll throw this question to Dr. Poland. How do you fight that dichotomy and effectively vaccinate people in a timely way while doing it in a personalized manner?
Yeah, Bob, this is a little bit of the Kuhnian dilemma of confronting new ways of doing things with all the objections, and that's a hard thing to do. I think first you develop the science, then you develop the strategies to implement that science. Maybe an example would be, I don't think you're practicing 21st century medicine if you don't genotype a patient before prescribing Plavix. And in fact, the FDA has put a black box around that. And yet most clinicians aren't doing it. And I think this is really no different. The other thing is we'll get to the point, I believe, where we work out the many, many factors involved in genotyping people perhaps at birth. Just as we now study them for some 30-odd metabolic diseases that are so rare, none of us know them all and have never seen them all, and then appeal to that genotype phenotype database that we'll have on every patient that we see.
Right. Chris Arisman has a question.
Yeah, I think I'm sort of following on your question, Dr. Hopkins, because I'm having been in the public health field, so having focused on population health my entire career, I'm trying to think about adapting in this paradigm shift and just wondering how we operationalize this in our field. So not saying can't, won't, shouldn't, but just wondering, how do we get more information? And I guess a specific question to this is just, if I showed up at Mayo Clinic, would that, and I needed to be vaccinated, would I be genotyped? And is that kind of the process that you use now? I'm just trying to wrap my head around this. Sorry.
Yeah, Chris, good to see you, by the way. No, that wouldn't happen right now other than under protocol. So, for example, we, like many institutions, are developing mega biobanks exactly in support of this kind of science, wherein we have cells, DNA, RNA, the clinical record, the clinical phenotype, so that we can correlate these things. It's a bit, and let me just take us back a couple, 15-ish years, a decade ago, where it was so easy for clinicians. You had in your refrigerator trivalent and activated influenza vaccine, and everybody got the same thing. Now, do I give LAIV? Do I give TIV? Do I give a recombinant vaccine? Do I give an adjuvanted vaccine? We're already doing it. We're already, in a sense, individualizing. That's not quite the same as personalized. That's the science is going to have to catch up to that ideal and then figure out, just as you're asking, how would we actually implement it? Well, in part, for example, my laboratory has studied different what are called TAP alleles in women who are HPV infected. And we've defined certain alleles where these women are never going to develop and never have developed cervical dysplasia or cancer. So the only reason to give them the vaccine is to prevent genital warts, not for the main reason that the vaccine was developed. It's just an illustration of the fact that eventually what will happen is clinicians will see a patient, and just like they have their past laboratory results, they'll also have their genotype flagged for alleles or SNPs of concern. Not there yet, but eventually.
Thank you. I won't drive down when I get back. So thank you.
Jewel Mullen has a question.
Thank you for a really compelling series of presentations, starting with Jim's comments on equity, getting us all the way to the realities that we know that not all vaccines work for all people. So building on the comments, as a public health person and primary care doctor, I'm sitting here thinking how we honor the 21st and further science at the same time that we're stuck in 20th century communication and faltering public confidence, not just among people, but populations, but clinicians who are parts of the people and populations as well. Because somewhere along this series of presentations, I heard lots of support for vaccine hesitancy, vaccine rejection, and pushbacks on the notion that someone might actually need to get vaccinated for something, in the same way that some people tell me they're not going to get or haven't gotten lung cancer yet, so they might as well not stop smoking. And as we do all of that and consider as well that even if we're thinking we have gene profiles on people, with the reminders of phenomena such as epigenetics, how do we answer and how should we start planning now to talk to people who want to know whether or not something will work for them and whether or not it will be safe for them? But sorry for the question, but it's what you got me thinking.
That's a great question, an insightful question, a question that deserves a thoughtful answer. So I'll give a try at the latter. Interestingly enough, when we first started publishing this, and David might have the same experience, the groups that contacted us with the greatest support were not clinicians or vaccinologists. It was groups and people I was able to identify in the sort of anti-vax and vaccine hesitancy spheres. So people who understood the power to say, wait a minute, this changes everything. If you're able at some point, and I'm not saying we're there yet fully, if you're able to say, this is a vaccine where we've tested this for you, and we know you don't fit into the category of people who develop side effect A. You're just not going to develop that. We don't see that in people who carry your HLA haplotype. And we know you'll respond. And by the way, you only need 2 doses, not 3 or 4. That is powerful to individuals, and I think may help. There's a lot of issues around vaccine hesitancy and rejection, but I think that would go a long way toward people's understanding and comfort level.
So following up on Greg's point, one way of thinking about this, sorry, this is David talking, is often what we're looking for many, ideal vaccine or immunization schedules is to actually reduce the dose, reduce this number of doses that are given, to ideally maybe give a vaccine once in a person's lifetime in a way that's mostly innocuous to them and they don't really have any adverse effects whatsoever. And so one way I think many researchers approach this type of question is to make the technologies work better and to try and really focus on those innovative approaches, and then to focus on the problems in a way that, okay, we solved one problem, let's move on to the next problem, right, Greg? That's kind of the way we try and think about it. And so that tends to be our way. Now, how we communicate that and how you deal with vaccine hesitancy in different ways may not always be transparent. But I think from the scientific and research translational aspect, that's how we tend to approach it, I think. And it sometimes aligns with the concerns of people who are vaccine hesitant, at least in some ways.
And I could offer as well, Jubal, the advance that we've seen in science and the types of opportunities demonstrated by the excellent presentations today really help us front load the ability to move new types, new paradigms for vaccine therapies into the clinic. But I want to reiterate the concern I flagged in my presentation that we need to move the entire ecosystem forward so that we can keep up with the speed of innovation. We need to move forward the pace of manufacturing. We need to move forward the pace of clinical research. We need to move forward the pace of regulatory science and regulatory review processes, because those are all things that have to happen between a good idea and the delivery of a vaccine to patients that need them. So when it comes to really studying more specific and more selective populations, it's absolutely the future. But remember that in so doing, we're restricting the size of the population that's available for study in clinical research. We're creating a more restrictive inclusion-exclusion criteria, and we're creating a much larger series of dossiers for regulatory science to try to decipher and decide on. So, we need to move all of those elements forward at the same pace in order to truly speed up the ecosystem.
And our final question or comment from Dr. Slater.
Thank you. Just to sort of reemphasize the concern of the frontline healthcare providers and the public health officials, these transitional periods are the most hazardous ones. And the attraction of personalized medicine in general is my ability to look you in the face and honestly say, no, you're not going to have an adverse reaction. Don't worry, we're good. That story is extremely compelling. The transitional story that says, well, we're going to be able to reduce your adverse reaction rate. We know that you have about a third the adverse reaction rate of the general population, so we're good to go, right? That's not going to work. For the vaccine-resistant population, as for all of us, we like stories, a narrative that goes from A to B to C, and statistics, stochastics doesn't work. So the transitional period between now to then is one in which we have to worry about the communications, because the communications are going to be as fraught as they were before, even if we're heading to a place where the communications are simple.
I want to thank all the members of this panel. I want to thank the members of the committee and our liaison and others that are on the call, as well as those in the meeting. We are now going to go to our lunch break. We will return at 1:15 p.m. Eastern Daylight Time. Thank you for attending our June 22 National Vaccine Advisory Committee meeting.
Produced by the U.S. Department of Health and Human Services.